Assalamualaikum and hi to Dr. Johari and to my lovely friends. Today we will present our fieldwork finding at Mersing, Johor. Before that, I would like to introduce our team. First, Hazel, Devi, Ada, Krisha and me, Batis. Moving to the introduction. Invertebrates are the most diverse group and animals that do not have backbone. Unlike vertebrates such as mammals, reptiles, amphibians, fish, and birds who have backbone, about more than 90% of all animals on the earth are invertebrate. Invertebrate can be found in both terrestrial and aquatic habitat, and it include ranging from coral, sea star, sponges, insect, worm, and crabs. Also, about 80% of all invertebrates are grouped with phylum Arthropoda, which includes centipedes, crustaceans, millipedes, spider, and insects. Fieldwork insects and invertebrates in the river or waterfall area and its surrounding were carried out to be identified the diversity of invertebrate or fauna in the area. The student will be provided with appropriate tool to do sampling of aquatic invertebrate. Appropriate tool like net, aquatic net, and using the stone shaking technique are used for sampling of aquatic invertebrate. Aquatic invertebrate sample caught should be kept in a container provided, and students have to identify the aquatic invertebrate. Also, students have to identify the important characteristic of each aquatic invertebrate by related reference material. Invertebrates are important to humans in many aspects. It is important in helping us in biological control of pests that can maintain the ecological balance in this environment. Also, aquatic insects play a vital role in the aquatic ecosystem. They help in breaking down and decomposing the dead material at the bottom of lake, such as dead fish, leaf, and other organisms. They also act as food resources for pork, fish, birds, and other animals. Aquatic insects are very good indicator of water quality in the lake or stream as they are sensitive to polluted water. Other from aquatic insects as bioindicator water quality are order Ephemeroptera, Helicoptera, and Trichoptera. Next is the objective of fieldwork. Number one, to understand the method of sampling aquatic insect and invertebrate. Number two, to identify abiotic factor and to know the appropriate tool to record parameter of abiotic factor. Number three, identify the important characteristic of each aquatic insect and invertebrate. Number four, be able to observe some aquatic animal invertebrates such as Porifera, Echinoderms, Cnidaria, mollusks, and arthropods. Number five, to identify and compare coral with coral in the Ecoma Mersing Johor. Last but not least, to recognize the habitat of some invertebrates near the muddy beach and classify them. Methodology will be explained by Krisha.
Next, we'll be looking at methodology. So the first thing here is the study area. Next slide. Okay, so in study area, we will be looking at three different places near Ecoma Mersing Johor, which are Pulau Batu Gajah, Hutan Simpang Gulang Arong, and also Tanjung Penyambung. Next, we'll be looking at the target population, where all of the invertebrate fauna that can be found, identified, and collected, which is more specifically invertebrates, mostly Porifera, Echinoderms, Cnidaria, mollusks, and also arthropods. Next, we'll be looking at the procedure. The first place will be Hutan Simpang Gulang Arong, where the sampling area consists of 4 different locations of sampling area, and we fix each place as 2 meters for the length of the river. Next, we'll be looking at the abiotic parameters, where we measure by using aquameter for temperature and the pH. The average temperature is 24.76 degrees Celsius and pH is 6.63. Whereas the light intensity is measured by using lux meter, where the average light intensity at the sampling area is 0.21 kilolux. And also we did measure the speed of river current, where we use the formula speed equals the distance over time. So the average speed of river current from two different regions chosen at random with 3 times repetition is 1.1 meter per second. Next, we'll be looking at the technique used, which are server net technique, rock shaker, and also aquatic net. So for all these three, we we use the stones within the net perimeter and rings in the net, and also the specimen that can be found are collected in specimen container. Next will be specimen identification, where all of the specimens collected were sorted out, grouped, and labeled according to their respective taxonomic groups and common name. Each group of specimen was stored inside multiple different vials containing 70% alcohol for preservation. And the next place is Tanjung Penyambung, which is a muddy beach area. And the specimens that are collected and also taken photo, they are also sorted out according to their respective phyla. And the third place is the Pulau Batu Gajah, where snorkeling activity is carried out and observation of specimens are also done. That's all from me. Next will be results and discussion.
Moving on to the results and discussion, the first phase is fieldwork at river of Hutan Simpang Gunung Arung. So these were the findings that we discovered. The first one is stoneflies. As you can see in the picture, there is a phylum of Arthropoda, class from Insecta, order of Plecoptera, family is Perlidae, and the characteristics are we found this Arthropoda from underneath brick rocks in the river, and they have distinct preferences such as specific diets, and they are also herbivorous, scavengers feeding habits, and useful for biological indicators, and they are incomplete metamorphosis, and they can be brown, gray, greenish in color. And next are true flies, which are the from phylum of Arthropoda, and the class of Insecta, order from Diptera, and the family of Muscidae. And the characteristics are it's found in the middle of the river, it's from the fast flowing water, and its diverse habitats, which are terrestrial, freshwater, such as marine, and it has variations of sizes. It can be found large and small. It's also a holometabolous metamorphosis, and it can act as a pollinators, predators, and also parasitoids. And true flies has a short lifespan. Next. Okay, the third findings at the river is maggots. So maggots are from the Arthropoda phylum and Insecta class, and the order is Diptera, and the family is Anthomnidae. And the characteristics for these maggots are it's found in the middle of river, it's legless, soft, and it lacks of exoskeleton, and it feeds on decaying matter such as rotting food, and it has a complete metamorphosis, and it has decomposition process and nutrient cycling. And for maggots, it's a rapid growth organism. Next. Okay, I'm passing on this caddisflies to my friend Balkis.
Next, we found a variety of caddisflies larvae. You can see on the slide there are 3 of them, which are with long anal prolapse, short anal prolapse, and brown color larvae. We found them on a rocky part of the river. Caddisfly larvae are adapted to specific mineral, water temperature, speed, polluted concentration, and sunlight exposure of that particular area. Each species of caddisfly larvae have their preference of different part of the river, diet, and water temperature. Different color part of the body show they are mainly herbivorous scavengers, and have different diet, which are plant material and dead organism. These larvae fall under class Insecta and order Trichoptera. Caddisfly larvae are sensitive to water condition. If the water conditions are favorable, then they will continue to develop through instar. Next. We also found mayfly named Canidae in the stream of the river under rocks. This species prefers slow-moving speak water where there is loose sediment and abundance of sleep. Their diet is organic matter or scraping algae that can be found on the rock and other substrate. Mayfly limbs mostly have two wing mouthpart, abdominal gills, legs used for movement like climbing and sprawler, and three tails. These mayfly fall under class Insecta and other form Ephemeroptera. They present in the river also show a good water quality indicator given their sensitivity to pollutants. Last but not least, we found the freshwater decapods, which are stream and freshwater crab in the area for our sampling. The stream is from the class Malacostraca. Many streams were found as our sampling area. It's considered as a place with low water current flow. They are important in controlling nutrient outflow and play a role in cycling nutrients from ecosystem. Shrimp have a variety of diet food which they are omnivorous. Meanwhile, the freshwater crab is also from class Malacostraca. Freshwater crab need pristine water condition to survive and a good indicator of water quality. Freshwater crab also omnivorous. They are feed on algae, dead frog, and fallen leaf. Crab play an important role in nutrient cycling in the freshwater ecosystem.
Okay, so now we move on to fieldwork at beach area of Tanjung Penyabung. Okay, so these are the results we obtained from our first day of field trip. We went to the beach area located at Tanjung Penyabung. We collected most of the invertebrates under the small and big rocks around the seashore. And the invertebrates that we found fall under phylum Echinodermata, Mollusca, Chordata, and Arthropoda. So for phylum Echinodermata, we found the sea cucumbers that fall under class Holoturoidea. It has a very soft and flexible body. The color of this invertebrate is darkish brown and it nearly resembles a cucumber. It has 5 rows of tube feet that it uses for locomotion. In addition, next slide. Okay, in addition, for phylum Arthropoda, we found baby crabs and barnacles, which are under class Crustacea. And for phylum Chordata, we found small tilapia under class Actinopterygii. Next slide. Okay, so for phylum Mollusca, mostly we found the class Gastropoda and Bivalvia. For class Bivalvia, we found a pen shell with characteristics of 2 thin walls and it has a fan-shaped shell which is dark brown in color. Pen shell is using the pointed bottom of the shell to attach to soft rocks, corals or dirt. Pen shells are frequently found buried in the sand with only top part of the shell visible in shallow seas close to seagrass beds. Next slide. Okay, next we found nesting mussels under class Bivalvia, even though it is in the same class with pen shell, but it has a few significant differences in characteristics, such as the shell has an uneven or wedge-shaped form. The exterior of the shell is typically blue or brown, giving them a dark tint. The insides of the shells frequently shines like pearls. The sensitive tissues inside mussels is supported and shielded from predators by their shells. Next. We also found a few invertebrates under class Gastropoda, such as orange slug, crown conch shell, tower snail, and also rock snail. These two snails are different in their shell shape, such as a tower snail has a cone-shaped shell, while a rock snail has a fusiform shell with a solid, thick, and sharply pointed backs. These invertebrates share a few similar characteristics. Next slide. Okay, these invertebrates share a few similar characteristics like having ventral and muscular feet and also secrete mucus on the ground for its locomotion. Besides, they also have 2 pairs of tentacles and also compound eyes. So since we collected more invertebrates from the class Gastropoda than from any other class, we may conclude from the invertebrates that we observed and collected at Tanjung Penyabong that phylum Mollusca is the abundant species in Tanjung Penyabong. That's all from me.
Okay, next, we move on to the third place, which we went to the Pulau Batu Gajah. The finding at Pulau Batu Gajah is mostly from the phylum Cnidaria, which is the class Anthozoa. We found the 4 species which we can compare to the Ecoma gallery. The first one is the Estrangia species. We can see the structure here is encrusting and branching. The second finding is the Archopora millepora. We can see here there is post structure scale on top of the body. The third comparison to the Ecoma is the Galaxia striata. We can see there is a star shape on top of the body, and common can see on the many southern shore. The fourth compared specimen to Ecoma is the Montipora hispida. We can see here the encrusting body, and also they are branching from the Ecoma specimen. We also found the specimen from the phylum Mollusca and class Gastropoda, which are sea snail. The scientific name is the Bucillenum species. The next one is the Nerita plicata, also the class Gastropoda. These 2 specimens is compared to the Google Lens, not to the Ecoma gallery, because Ecoma gallery mostly have the coral specimen. Also, we found at Pulau Batu Gajah is dog conch seashell. The scientific name is the Levistrombus canarium. And we also found the Echinolithorina malacana. We can see the difference between dog conch seashell and sea snail, the color and the structure. For this Echinolithorina, we can see the structure is wrinkled, and the seashell is yellow colorish. And the 2 last one, we found the wedge clam from the phylum Mollusca under the class Bivalvia. The scientific name is Donacilla cornea. And the last one is the sea mouse from the phylum Annelida. The scientific name is the Aphrodita species.
So, as a conclusion, the aquatic animals in vertebrates, mostly porifera, echinoderms, cnidaria, mollusks, and also arthropods have been well observed with the group members through their unique features, and several abiotic factors have very significant impacts on the survival and the reproduction of the species in the ecosystems. So, the study of invertebrates has proved their existence as the bioindicators to the ecosystem and the keystone species that helps to define an ecosystem. Thank you for listening.